Pleased to welcome you to the second webinar of the BIOCON-CO2 project and webinar series in general on bio-CCU, carbon capture and use. Today's topic is CCU biocatalysts, how to get them out of the laboratory. I'm Andreas Schweinberger. I'm working for the company ARTTIC in the Berlin office, supporting stakeholder communications. That's also the reason why I give this introduction to the webinar today, supported by my colleagues Suzanne Barrault and Sarah Kruxater from the French branch of ARTTIC. So my honor to welcome you and to open this webinar. I give a few, as said, introductory slides, and then hand over to the BIOCON-CO2 scientific coordinator that you already see in the live stream, Annabel Serpico from LEITAT, who will then moderate the presentations and discussions. For today, we had received 70 registrations to a large extent outside the BIOCON-CO2 project. So if we are now at 43, I mean, this is a nice echo, and we're happy that you found your way to this online event in these extraordinary times. And I want to come from projects that collaborate within the Cooling the Earth initiative, and who may have spread the word about this webinar. Before we start with today's content about biocatalysis, just a short reminder what the purpose of this webinar is. This slide just summarizes in a few bullets. It's not a classical teaching event as you may expect from the word webinar. We had several summer schools and scientific workshops that cover this extensively. Here we are more focusing on raising important issues about biological carbon capture and use, and in particular to do this in the framework of a platform for discussions, which is open not only to scientists, but to also connect to other concerned stakeholder communities. I think of in particular of industrial users, but also regulators, policies, society at large, and to somehow connect these people through these meetings, and in an ideal case create a kind of information sharing wider community. And of course, as all the EU projects are supposed to do, to promote their topic, which is like bio-CCU and the project results per se. Could you move on to the next slide? Okay. So as we speak of a series, in total we plan to have 4 events. The first one took place in early December, focusing on bio-CCU in general, and including its potentials and opportunities. The second one, as of today, is, as said, on CCU biocatalysis and how to get it out of the lab. Maybe a few words on this, just to highlight also what has been said before about this webinar. To mention what the goals of today would be, it's of course first of all to give you an overview of what we understand by biocatalysis, and make a short stock take of the ongoing R&D by the different experts that you see already here with their live cams and their presentations, and to get a better overview and understanding. Then raise awareness of issues that still hamper or block industrialization of biocatalysis, which still remains contained at the lab level. And then discuss with you, first of all, with the panel members that you see already, but then later on with everybody who feels like raising the voice, what research can do to meet the demands for upscaling and industrialization of this technology. Then 2 more webinars are foreseen. No date so far. Usually we thought to have them in 3 to 4 months intervals, but as the project has been extended as to many due to the COVID situation, this is still to be sorted out. Anyway, registered participants who agreed that we can use their email addresses will receive email invitations to upcoming events. Could you move on to the next slide? Okay, just briefly about the logistics and code of conduct. So this webinar, as you may have recognized and be informed, is recorded and will be made public via the project website. All attendees are muted for the time being. We'll start with a set of presentations first, as you may have seen in the agenda. Please feel free to ask questions by using the chat window. And we'll take a few questions after each presentation if there are. Then later on in the final 30 minutes, we plan to have a panel discussion, and there we will open also the mics, and there you can make your comments or ask your questions directly to the panelists. Please, as usual in this kind of online events, raise your hand before, and then don't forget to unmute yourself before you ask. Next slide, please. And here you see the overview and a short presentation of the speakers and panelists of today's webinar. As said, Annabel Serpico, the technical or scientific coordinator of the BIOCON-CO2 project. She's a researcher in applied microbiology and biotechnology. Tom Ewing is scientist at the Wageningen Food Biobased Research. He is interested in the development of biocatalytic processes for the production of chemicals and materials from renewable feedstocks. Marco Fraaije, I hope I pronounced this correctly, is full professor at the Rijksuniversiteit Groningen. He's working on enzymology of redox enzymes, enzymes discovery and engineering, and biocatalysis. Followed by John Woodley, who's a professor at the Danish Technical University. I think this is the right wording for it, in the chemistry department. He's specializing in biological synthesis of useful chemicals using biocatalysis and fermentation. Last but not least, Albert Carceller from the Universitat Autònoma de Barcelona, a PhD student in applied biocatalysis and bioprocesses engineering. Then on my last slide, you get an overview of the agenda or program for today. First of all, Annabel will give us an introduction to the CCU and in general to CCU and the BIOCON-CO2 project results in this domain so far. Tom will give us an overview of biocatalysis for CO2 conversion in general. Marco will speak about generating stable enzymes by computational enzyme engineering. John will show us the industrial requirements for translating lab-scale reactions into industrial bioprocesses. Very interesting contrast to the scientific talks before. And Albert Carceller from the Universitat Autònoma de Barcelona will speak about CO2 valorisation, multienzymatic synthesis of lactic acid using CO2. And then Annabel will lead the panel discussion and close this webinar. So Annabel, are you ready? I hand over the actual presentations to you.
All right, thanks Andreas, thanks for the introduction. So as said, I'm Annabel Serpico and I'm the current scientific and technical coordinator of the BIOCON-CO2 project. And I've decided just to start by giving you an overview of the BIOCON-CO2 project, the aim, the purpose, so that will give a bit of context to the webinar of today. So if you please move to the next slide. Thanks. So BIOCON-CO2 project is Horizon 2020, which aims to transform raw CO2 from iron, steel, cement, and electric power into added value chemicals and plastic, with the purpose of reducing greenhouse gas emissions and by developing a flexible platform which aims to transform raw CO2 into the targeted product. that has the purpose to increase the sustainability of the chemical industry and as well support European leadership in biological CO2 reuse technologies. So as said, it is an Horizon project, lasts 4 years. We were recently granted, as Andreas commented, an extension of 6 months. So we are still like have a year and 3 months to go. And it's coordinated by LEITAT. Next, please. Next, please. Okay, perfect. So the BIOCON-CO2 platform aims at developing this flexible platform via 3 main approaches. So the improvement of CO2 solubilization via a series of approaches such as reactor design, deployment of enzymes, and improvement of reaction conditions. The development of several bioprocesses and the improvement of those bioprocesses via genetic engineer, metabolic engineer, enzyme engineer, and last but not least, downstream technologies to maximize not only the product formation, but also their recoveries. So these 3 main platforms combine all together as puzzle pieces and aim to develop 4 main microbial cell factories: aerobic, anaerobic, whole cells biocatalysts, and purified enzyme, with different targeted compound and different end user target industry. So we have C3, C6 alcohols, poly-3HP, and formic and lactic acid. Next, please. We have a very nice consortium, 18 partners spread around 8 countries with recognized industry expert and leading academic organization. And we have a very nice widespread between large industries, as we have 5 large industries, 5 SMEs, 4 research and technology organization, and 4 universities. Next. So the expected results that we aim to achieve are the assessment and validation of low energy microbial processing system able to convert CO2 into the targeted product, and by producing 4 chemical building blocks, and scale up these technologies developed within the project at a pilot plant level, so whose construction is ongoing, and it's aimed to be achieved by the end of this year, and improve as well the public perception of CO2 reuse technologies. So this is a very broad overview of what we are trying to achieve in this very challenging project. So please follow us also on Twitter or the website. They are daily updated. If you want to hear more about the achievements, I think this year, which is our last year, is going to be full of nice results. So next, please. Without further ado, I'm just going to give you an overview of the topic of today, which concerns CCU Biocatalysts. So I'm just going to set the scene for the next speakers, giving you just an idea what's the current situation. So carbon capture and utilization, I think this is a concept that you all are aware of. It's the concept of utilizing, not just capturing, but utilize CO2 and converting directly into valuable molecules, contributing, of course, significantly to the CO2 mitigation. Next, please. This does require like a mindset switch, so just not thinking anymore as CO2 as a pollutant, but actually as a valuable feedstock that has several advantages. First of all, it's potential of being converted in a variety of molecules. You can see on the left-hand side the possibility that CO2 gives you, and it does have several advantages because it's a very widely available resource. I think it's one, if not only the most abundant in Europe. It's sustainable as it's outside the food chain, and its deployment would, of course, reduce and avoid at the same time CO2 emission, contributing to a low carbon economy. Next, please. different strategies have evolved to utilize CO2 and convert them into fuel and chemicals. In particular, biological approaches have drawn attention, and I think there's lots going on, and there is a lot of interest of using that for the production of chemicals of interest. So biocatalytic process, what are those? They involve the synthesis of chemicals using biological systems. They have several advantages compared to the chemical counterpart, as they are safe, not toxic. They have very high regio, chemo, and enantio selectivity. They do require very mild conditions, so general ambient temperature, atmospheric pressure, neutral pH, and they are, of course, environmentally friendly. However, they do have some cons. Probably today we are gonna talk a lot about the cons and how we want to overcome those limitations. And they include the sensitivity to several factors such as toxic substances, temperature, as well as very often the requirement of cofactor regeneration systems. Next. Amongst the biocatalytic CCU, we can distinguish between microbiological route and enzymatic route. Next, twice, so you have a perfect. So with the microbiological route, we include the microalgae and bacteria one. I'm just gonna probably focus more on this part, as Tom afterwards is gonna talk much better than me on the whole enzymatic part. So amongst the microbiological one, we distinguish mainly between, as I said, microalgae and bacteria. And I'm gonna give you a few examples, you know, higher tier of the application of these microorganisms. The advantages are clear, especially for the microalgae, because they have very high photosynthetic efficiency, fast sequestration rate. We have several examples of co-production of food, feed, fuel, fine chemicals, and however, they do require like very large lands, so that has limited their application, especially in Europe. As for the bacteria, they have a faster life growth cycle, and they rapidly adapt to the changing environment. However, they have low volumetric productivities, and that has been the main drawback as well for this kind of application. As for the enzymes, they easier to produce. You can control exactly the reaction as when you're having a much more complex system. In case of microorganism, is not as straightforward, and you can improve it, and you hear a lot more about that from Marco. You can improve the enzymatic performances, the stability, and you can target at that. However, as mentioned before, it usually requires cofactor regeneration systems, and usually this increases the cost, and so you have a very high cost and low productivity. Next. So I said before, I decided just to give you two examples of some of technologies that have reached a higher TRL, which have demonstrated the feasibility of these technologies. So as for the microalgae, I picked the Solix, which is a US company which has developed photobioreactor system for the production of algal biofuel. You can see that they have a very large demonstration plant based in Colorado, which has reached over 3,000 gallons of algae oil per acre of product in 2010. So that demonstrates that this kind of technology is possible. However, does require very large lands. Next, please. As for the bacterial CCU, I picked this example, which I think is very closely related as well to what we are trying to achieve in BIOCON-CO2, and is the LanzaTex technology, which produces ethanol from gases from steel mills. So how does it work? So basically the gas is cooled down, cleaned, and fed to the acetogenic microbe, which then produce ethanol, which is then separated from the cultivation broth and recovered. So this technology has been demonstrated in several plants worldwide. So we have it in China, in the US, in South Africa, and also in Belgium. Next. So we have a very wide array of microbes that could potentially contribute to the production of an array of products. However, this has still been limited. The main limit is, well, low yield and high production cost. So that has limited the wide, well, which is not, yeah, just to summarize it. So what we have to do in order to target the widespread of biocatalytic CCU, next please, will be two main challenges would be to increase the productivity and reduce the cost, because while the current commercial facilities demonstrate the feasibility of the biocatalytic carbon utilization, the technology must be deployed more widely and at much larger scale in order to have also significant impact on the carbon dioxide sequestration. So in order to achieve that, the conversion must increase to reduce the actual cost. And as well as a broader range of product must be targeted in order to further impact the atmospheric carbon levels. And so we would have to increase the product portfolio of the biological CO2 conversion that are available right now. So without going further, I think this has given a bit the overview of what we are going to discuss today. So I'm just gonna hand the word to our next speaker. Next, please. So, which is Tom Ewing, is one of our partner in the BIOCON-CO2 project, and is going to give an overview of biocatalysis for CO2 conversion. Thank you.
Yes, thank you, Annabel. My name is Tom Ewing. I work as a scientist at Wageningen University Research in the Netherlands. And I specialized in biocatalysis, so the use of enzymes for chemical synthesis. And in the BIOCON-CO2 project, Annabel already introduced a work on using enzymes for carbon dioxide conversion to chemicals. And in my talk, I'll give an overview of what is possible in this field. So today we're talking about bringing these biocatalysts out of the lab, but first I will introduce to you what is the current state of affairs in the lab. So what can we actually do at the lab scale with this type of enzymatic catalysis? Can we go to the next slide, please? So Annabel already really introduced why we need to, or what the advantages are of converting carbon dioxide to chemicals for carbon utilization. So I'll limit myself to saying that there's a lot of carbon dioxide emitted to the atmosphere, and it would be great if we could do something useful with this, for example, converting it to chemicals. And one of the ways that we can achieve this is through enzymatic catalysis. And I will introduce you to the chemistry that can be performed in this way. So if we can go to the next slide, maybe. So if we look at the possibilities to use enzymes to convert carbon dioxide into chemicals, we can really define two main types of reaction that we can perform. The first is the reduction of carbon dioxide to one carbon atom building blocks. So this is sequentially the reduction of carbon dioxide to formic acid, formaldehyde, and then methanol using 3 separate enzymatic steps. And in particular, methanol is a very large volume bulk industrial chemical. The other type of reaction that we can define that we can perform enzymatically to convert carbon dioxide is a slightly more complex chemistry, to put it that way. So this is actually carboxylation reaction, so where we use carbon dioxide as a carboxylating reagent, for example, for phenols, or we can carboxylate also acetaldehyde to pyruvate, a precursor to lactic acid, which will be the topic of one of the other talks later in the afternoon. But I will introduce you briefly to these two different types of reaction and what are the pros and cons of them. So if we go to the next slide, please. So in terms of the reduction of carbon dioxide to 1 carbon atom building blocks, I already mentioned that this is a 3-step sequential reaction system we go through formic acid to formaldehyde to methanol. What are some of the advantages of doing this enzymatically rather than in a different way, for example, chemically? Well, we can think of the selectivity of enzymatic catalysis. So if we want to make sure we only really get one reaction product. Also, the reaction conditions under which these enzymatic reactions occur are quite mild, which is an advantage. There are also some challenges associated with these reaction systems. One of them is that we need a source of electrons in order to perform the reaction. So we're doing a reduction reaction, means that we need to get electrons from somewhere, and this can be a challenge, which I'll discuss in a bit more detail in the rest of my talk. Another challenge can be the stability of enzymes under industrial conditions, and also increasing the productivity and the yield of the reactions to the amount that is required for an industrial application. Now I want to introduce you to some of the electron donating systems that we can be used for this type of chemistry. So if we can go to the next slide. So one well-established way of donating electrons to carbon dioxide with this kind of, to carbon dioxide with this kind of chemistry is the use of a second enzymatic system, which makes electron transfer possible. So this is some work that was done in our department some years ago, where we worked on a system to convert carbon dioxide to methanol. And here we use enzymes that are dependent on a cofactor, an NADH cofactor, which is a direct electron donor to the enzyme for the reaction. Now this is of course nice that it works with this electron donor, but a problem is that this NADH is expensive, so we cannot use it in stoichiometric amounts during the reaction. We have to recycle it in order to be able to generate a cost-effective system. And here we used a method of recycling this NADH based on using a second enzyme, a glucose dehydrogenase enzyme, which converts glucose to gluconic acid, thereby regenerating the NADH. So this is one system that can be used as an electron donating system. It's well established, it works well, but the disadvantage of this is of course that we get a second product, the gluconic acid, that we have to separate out. And in the end, if you want an industrial process, you also have to find an outlet for that. So that is something that's less advantageous. Can we go to the next slide, please? Some other systems that can be used for electron donation are electrochemical systems. So this is where we have an electrode and supply electrons to it, which are then usually via a mediator molecule transferred to an enzyme or the enzyme's cofactor. This has the advantage compared to the system I showed you before, that we don't necessarily get a second product of the reaction. There's still challenges in terms of the example I give here. They use not an NADH dependent formate dehydrogenase, but a metal dependent formate dehydrogenase, which can be relatively easily electrochemically reduced. But these enzymes are not really stable enough for practical applications. There's a challenge there in this case. A third example of a method you can use for the electron donation is, in this case again for the NADH dependent enzymes, is to use a photocatalyst to donate electrons. So there we have a catalyst which, when irradiated with light, will donate electrons again from a second substrate, in this case glycerol, to an NADH cofactor, which can then be the cofactor of the enzymatic reaction. This again has the disadvantage of forming a second product. Can we go to the next slide, please? So in the BIOCON-CO2 project, we are working on this kind of chemistry for the C1 the reduction of carbon dioxide to C1 chemicals, where we're using a system for the production of formic acid from carbon dioxide based on the use of a formate dehydrogenase enzyme, which catalyzes reaction using the enzymatic system with glucose dehydrogenase and conversion of glucose to donate electrons to it. And we're working on developing a robust and efficient enzymatic system for this, and also optimizing the process with the idea that in the end we will apply it to gas mixtures that mimic the composition of industrial off-gases from for example, the steel industry, to show that we can use this type of system to convert carbon dioxide from, let's say, realistic gas streams. Can we go to the next slide, please? Okay, I just briefly want to also introduce this example of carbon dioxide conversion chemistry using enzymes. So where there's a slightly more complex, let's say, chemical reaction, and this is the carboxylation of phenols using the enzyme known as benzoic acid decarboxylase. So where carbon dioxide is introduced into the phenolic group, and it's really an analog of a chemical reaction known as a Kolbe-Schmidt reaction, which can also be used to obtain this kind of molecule. Now this is actually quite selective, so that's not the main advantage of enzymatic catalysis here, but an advantage is that we can do it at far lower temperatures, so at milder reaction conditions, and you can do so chemically. And if we go to the next slide, and here we can see that this type of enzymatic carboxylation reaction can be used to carboxylate quite a wide range of phenolic compounds, and this can be interesting because this type of polyphenolic compound can be interesting due to their antioxidative properties, so it can be interesting to be able to modify them. If we go to the next slide. This is my final slide, so just a summary. I hope that I've shown you that enzymatic catalysis can be used to convert carbon dioxide to one carbon atom chemical building blocks, but also for more complex chemistry to carboxylate, for example, phenolic moieties. Advantages of biocatalysis for this type of reaction include selectivity of enzymatic catalysis, also the mild reaction conditions that enzymes are active under. Some challenges that there still are in order to optimize these systems and bring them hopefully eventually to industrial application include tuning the enzymes, improving their stability, improving the productivity of the reaction systems, and then specifically for the one carbon atom chemistry, developing efficient systems to donate electrons to the enzymes that are cofactors. So with that, if we go to the next slide, I would like to wrap up and thank you all, and then hand back over to Annabel, I guess.
Thanks, Tom. Thanks a lot. So I think you're setting perfectly the scenes for our next talk, which will be by Marco Fraaije, which is a leading expert in enzyme engineering and application in biocatalysis. Thanks. So I'll give you the floor.
Yeah, thank you, Annabel. Welcome all. It's a pleasure to present this afternoon and discuss this afternoon anything related to what I will present and what the other presenters have presented. My name is Marco Fraaije. I work here at the University of Groningen, leading a group where we do a lot of enzyme engineering. And in BIOCON-CO2, we have a role in engineering the enzymes that we see as interesting candidates for the project. And as Annabel and Tom both have already suggested, one of the challenges of using enzymes in industrial applications is their stability. And that's why we spent quite a bit of time in the last years to develop computational tools to come up with reliable tools to engineer stable enzymes. Next slide, please. So to discuss the engineering, I first should go to the properties of enzymes. And here on this slide, you can see a picture of a cell, which is the typical environment where enzymes are. And realizing that enzymes are in cells, you also can conclude that enzymes are used to mild conditions, neutral pH, not so extreme temperatures, aqueous solution, though not really aqueous. You could say that enzymes don't want to be alone. Enzymes are used to be, well, surrounded by all kinds of other proteins, biomolecules, DNA, pigments, and so on, as you can see on the picture on the right. And that also explains that if you take enzymes out of their natural context, they often don't behave that well. They are in an environment that they are not so stable at. And anyway, nature has never had never the intention to make enzymes that last forever. They should be recyclable in cells to regenerate other proteins. So that explains why enzymes in practice for industrial applications suffer from stability issues. Still, we know that enzymes can be used, but often you need to improve the stability. Next slide, please. So on the next slide, you see, well, some of the strategies that can be taken to make more stable enzymes. And often you need enzymes that can tolerate better the solvents that are used in a certain process, or they have to perform for a longer time. So during the last decades, you could say, quite a number of approaches, strategies have been evolved to come up with solutions for this, for getting a more stable enzyme in a process. And the first strategy mentioned here is related to physical methods, you could say. People have found that by, for example, immobilization, when you couple your enzyme to a carrier, to some solid material, you often find that the enzymes are more stable. That is probably partly because they are now already a little bit more like in a natural environment, surrounded by other proteins. So that's one way to go, but it's not always working for every enzyme. Other physical methods are encapsulation. There is quite some development there nowadays, that you can put enzymes into small vesicles or protein capsules. And we also have explored this a little bit. So for the ones that are interested, I put a reference there for a recent paper on this approach. Then what has been popular has been the directed evolution, developed not too long ago, which works in quite some cases, but it's a method that is quite laborious. So not so sure whether it will stay popular in the coming years. I would say the last two strategies here on this slide, they seem to be very promising. They have achieved quite some good results in the last few years. The first one is the sequence-based approaches, where nowadays you can use sequence information, so not even structure information, but just based on a protein sequence. You can do analysis such that you can predict what mutations you should make in a protein to make it more stable. Or you can even go for resurrecting ancestral enzymes. It has been found that if you calculate, let's say, the sequence of enzymes in the past, which is nowadays possible, you often end up with a more stable variant. Again, I added a paper there that may be of interest if you're interested in this approach. For today, I focus more on the last strategy indicated here, and that is where you use computational predictions that are based on structural analysis. So based on enzyme structures, it has become possible to do at least reasonable predictions on what you should do to improve a protein stability, enzyme stability. So this is only feasible since, let's say, about 10 years or so, but year by year, these approaches improve in quality and are nowadays, I would say, the preferred way to go to engineer a stable enzyme. Next slide, please. So there is no single solution and no single protocol at the moment that is really the best, I would say. By now, there are a number of approaches by which or in which computational power is used to predict mutations, so changes that you should make in a protein structure to make a more stable protein or enzyme. And I just mentioned a few here, a few servers, in fact, that you can consult on internet, where you only need to put in, well, your enzyme, the name of it or the sequence of it, and these servers will do predictions. They will generate predictions on what changes you should make in your enzyme to make it more stable. Also added here a reference that is more complete in the website servers that are available by now. And all these approaches have, well, one thing in common. They need a protein structure to do the calculations, but they all have their own protocol, their own approach to calculate what is best to do to make a more stable variant. So, for example, SS bond pre is the server that will tell you where perhaps a disulfide bond will improve the stability. And the other servers will be based mainly on one approach. Attractive here is that these servers are quite quick. You get your results in a few minutes or hours, but that's still all results on paper. You could say, yeah, this is just a prediction what you should do, but you still need to do it in a lab. And then one has to wait and see which one really works. And the predictability, I have to say, well, it's not always perfect. And therefore, in our group, we focused, we focused on combining some of these kind of approaches into one protocol, such that the predictability at least is getting to a reasonable predictability. So for that, I have a few slides more. So please, next one. The protocol that we developed is called Fresco, and I would like to go through this protocol step by step on this slide. As with the methods I mentioned before, in this protocol you need a structure to start with of your target enzyme. Well, nowadays there are hundreds of thousands of protein structures, so now it's not so uncommon to have a structure of an enzyme. And with that, we have calculations done, mainly using Rosetta and Foldix packages, but we can also include disulfide bonds if desired, or consensus sequence analysis. And this is all done in the computer. So then in a couple of days, the computer spits out, well, a few hundred of mutations that are probably going to give improved variants. And this is, in fact, a small section, a small selection of all possible mutations that have been calculated. Because the nice thing here is that in this approach, every possible mutation in a protein structure is analyzed. Well, in a lab, that would never be possible, or at least it would take ages. But that's the advantage of using computers. You can do calculations on any possible mutation. Well, computers are still not perfect, or at least the programming is not perfect. You still need visual inspection. That is the third step here. So it takes days to go through all the hundreds of predictions and select the best ones, at least based on our visual inspection. And then the lowest, on the lower part of the slide are the experimental steps. So then we typically take 100 or 200 mutants. We make them in the lab. It takes a week. We screen them also in that same week for thermostability. And then there is this nice additional step. We can then decide to combine mutations, because all these predictions are based on single mutations that in principle can be combined to combine the beneficial effects. So that in total, in this procedure, that takes, well, if you have everything in place, about a month or two, you will end up with a more stabilized enzyme. Now, to illustrate how this pans out, I have a few more slides on one project that we did on an alcohol dehydrogenase. So please, next slide. An alcohol dehydrogenase was studied where we found that this bacterial enzyme was quite an interesting one, expressed very well, was active on interesting alcohols. But one problem was that it was not so stable. the melting temperature, the stability was up to about 40 degrees. We were lucky enough to get crystals, and that we could obtain a crystal structure, and with that we could do the fresco approach. So on the next slide, you see the results we obtained from the fresco library that we made. So we made 177 single mutants, which can nowadays be done in a single multi-well plate, and we screened those mutants for stability and activity. And half of the mutants were indeed more thermostable, so the upper half, and we selected the number of mutants there that were also active to combine. And on the next slide, you can see what is the result of that. So if you take these single mutants, so that's the left half of this graph, you could say, they all have beneficial effects up to from about added a few degrees improvement up to a bit more, 6 degrees improvement. Individually, that's already nice, but then if we combine them all, which is the M10 mutant, you go from a stability that goes up to 43 degrees to 94 degrees. So that's quite a spectacular improvement of the thermostability of this enzyme. And that's really nice, because what we also observe typically is that if you improve thermostability, you also improve, for example, solvent tolerance. Next slide, to illustrate that this generated mutant, and here I have the data of the M9 mutant, is really performing better, also with respect to conversions. On the left, you see the rate of catalysis that at, well, let's say, lower temperatures are similar to the wild type. But then if you go to higher temperatures, well, the wild type just dies. But with this mutant, you can go up to 80 degrees and it's still fully functional. So it's doing well in catalysis and also in storage. So on the right side, you see a graph indicating that this fresco mutant can be stays stable and active for days, while the wild type, the original enzyme, degrades within a day. So this is as an illustration how a computational tool works out in this example. Next slide, please. We have tested the protocol that we call Fresco also on other enzymes, so that can be seen on this slide. So more than 10 enzymes have been tested so far with success. As you can see, well, the example that I showed is the best so far. So I have to be honest on that. This was the best example, but at least you see that what can be done. But also for the other examples that are in this bar plot, they could be improved significantly. We learn more and more by doing more of these kind of improvements with different kind of proteins. For example, now we also seem to see a trend that the higher oligomeric structures you have, that are these blue dots, the more chance you have to improve the thermostability. So if it's a tetramer, like this alkyl dehydrogenase, it seems to be easier to improve the thermostability. And there may also be other factors playing a role. For example, the ones that were not very successful, so only 10 or 15 degrees improvement on the left, they are all cofactor containing enzymes. And that's, well, we think to know why that is still not the best solution here, though still there is improvement. I hope with this you get an impression of what is feasible with computational tools, in this case specifically Fresco, but as I introduced, there are also more methods that can give similar results. And I think that the bottom line is that computational tools are improving every year again, and I foresee that certainly stability engineering is a doable thing for almost every enzyme in the near future. Please, next slide. I think it's the last where we can close this contribution for engineering stable enzymes, which just gives the picture of a resurrected ancestral dinosaur, let's say, with some group members of my group. Well, that's it for me. Please put in any question you have in the chat, and I will answer them now or later.
Thanks a lot, Marco. I think that's been a very nice overview of the tools available to improve the enzymatic stability. So without further ado, I'm just going to introduce the next speaker, which is John Woodley, professor at DTU. I just going to pronounce the acronym. So, which is going to give, I think, a very interesting overview and a different vision on how to scale up the reaction at industrial level. So, thank you.
Thanks a lot, Annabel, and thanks very much for the invitation to come and speak here this afternoon. I think this is a super interesting workshop, especially because we talk about moving things out of the laboratory. And as a bioprocess engineer, originally a chemical engineer, that's what I'm most interested in doing, trying to really get things out of the lab and start to scale things up and to really run them as an industrial process. So I'm based at the Technical University of Denmark, DTU, just outside Copenhagen. And I lead a group there which is interested in all aspects of scale up, also in some stability issues. As Marco has just mentioned, stability is often very important in the scale up of these. But I'd like to start by discussing a little bit some of the challenges as I see, which are not dissimilar to what Marco has said, but I think these are very important things to think about. So can I have the next slide, please? So this is a picture of what we really talk about. Yeah, when we're going to get out of the lab, we talk about making big scale processes. This is a tank, a 400 cubic meter tank running in United States in an industrial bioprocess. It's running a different type of reaction to the reactions we're interested in here. And this is in the process of them building the factory. And I put it here just to give you an indication. There is a man at the bottom of this slide, maybe you can see that, just to give you an idea of the scale that we talk about. And CO2 processes are surely going to be at a very big scale. So that's also something to try to bear in mind. But what we talk about then is that when an enzyme or a cell is inside this tank, of course, the conditions throughout this tank are not going to be the same everywhere. And that's part of the very important lesson about trying to scale up and to go to a larger scale. Can I have the next slide, please? So just as Marco, I also think that the challenge here is that enzymes and cells, of course, what they see inside, or what an enzyme sees inside a cell, what a cell maybe sees in nature, is very different to what we require in an industrial reaction. So the first very important message is that the conditions are very different. And that means whatever we screen for or whatever we look for in the laboratory has to be under conditions which are the same as those, or as close as we can get to those which we see in an industrial process. So there's quite a shift here from the lab scale to the industrial scale. Next slide, please. I've started to list here what I think are some of the potential industrial reaction conditions. So just as Marco perhaps put a little bit more emphasis on what happens to an enzyme inside a cell, I'm putting a little bit more emphasis on what happens when we're inside the scaled reactor. So first of all, we have high concentrations of substrates and product. And I mean by high concentration, somewhere between 50 up to even 300 grams per liter. It depends on the application, it depends on the prices and the economy, but it gives an idea of the type of concentrations of substrates and products we talk about. Bearing in mind they can also affect, of course, the stability of an enzyme or a cell as well. High salt concentrations, organic solvents, if we need to solubilize some substrates. We may have a liquid-liquid interface if we have the organic solvent beyond saturation concentration. It might be very useful for carrying certain substrates or products. There are industrial processes which operate in that way. And then gas-liquid interface for reactions which consume oxygen or produce CO2, or of course here when we talk about CO2 capture and use, exactly as Tom has mentioned earlier, then we really talk about using CO2 and getting it into solution, of course, is very important. But here we also have a gas-liquid interface. And many times we see proteins do not like to be at a gas-liquid interface. and that they start to unfold, or in the case of multimers, then their subunits fall apart. And this is really a challenge as well, to be able to deal with that kind of environment. And then the final thing I put down here is heterogeneity of temperature and pH and concentration throughout this huge tank. So in that 400 cubic meter tank I talk about, of course, it takes some minutes for material to flow around the tank. Conditions at the bottom are not the same as conditions at the top. So these are also things which need to be taken into account. Next slide, please. Over the last few years, one of the things that we've been doing in our group is to try to think about a procedure to help with this translation of scale from the laboratory to a larger scale. And one of the things we've started to identify is that it's a kind of a three-step procedure. And actually that begins, first of all, by looking at the biocatalyst. In an engineering sense, the biocatalyst is also, and changing the biocatalyst is a kind of degree of freedom that we have in our system. Other types of chemical system do not have that there. So we are very fortunate in having that, this ability to be able to change enzymes in the way Marco just brilliantly described is one of the great things we really have to play with here. So biocatalyst performance has to be correct. Then we need to think about the reactor type, and finally about the process configuration. So I'd like to spend a few moments just talking about each of those different areas now. Next slide, please. So in terms of identifying the performance, I of course want to have a way that I can measure the performance of my biocatalyst. And to do that, we're using what we refer to as metrics. These are very simple ways of being able to get an idea of how well the process works. This is in relation to the type of costs that we expect to see in the process. So without doing a detailed economic analysis, we get an idea by using these metrics what the kind of cost is and whether we're in the right ballpark to really start to think about scaling up the process. These metrics for biocatalyst performance, there are 4 of them for an enzyme-based process. So a metric for substrate cost is reaction yield, grams of product per gram of substrate, or maybe mole of product per mole of substrate if you prefer. I think everybody knows about that, but what people don't always know about is the biocatalyst cost. And here the specific yield is what we're using to measure that. So how many grams of product I get per gram of catalyst. There are people that are more interested in using total turnover number, for example. That's a similar kind of metric, but it's also important to realize that when we buy enzymes for industrial use, they're not always pure. And so having this kind of more practical metric here of grams of product per gram of catalyst is particularly useful. For downstream costs, we can think about also the cost in terms of the concentration which goes into the downstream, so the titer. And then the metric for the reactor cost is the rate or the productivity. And there's a trade-off between this specific yield and the productivity. In other words, I can make my reaction go faster, I put in more enzyme. But eventually, of course, that's also going to cost me something in terms of the extra enzyme I have to add. So that's the limitation, if you like, concerning the specific yield. So this trade-off is kind of important, and the specific yield needs to be measured over the whole lifetime of the biocatalyst. And that's one of the challenges, how to be able to do that in an effective way. Next slide, please. So a simple algorithm could look something like this. We define the added value of the reaction based on the value of the substrate and the value of the product. Obviously, the product has to have a higher value than the substrate. I think for CO2, that's not hard to do. But then we also need to think about the yield as well. So how many grams of product we get per gram of substrate. And this in the end will reflect the potential profit we can get from our process, or if you like, how much money we can put into the downstream processing and the other aspects of the conversion. We define some targets then. We can define targets for a specific yield reflecting the allowable biocatalyst cost, for product concentration or titer, which reflects the allowable downstream processing cost. and then the rate or productivity, what chemical engineers sometimes refer to as space-time yield, in, if you like, grams per liter per hour, and this reflects the reactor size as well. We define those targets, we try to make measurements about where we are in the laboratory today, and the gap between the two defines the enzyme engineering that is required. So it gives targets directly to people like Marco to say, okay, these are the kind of improvements which we require in the biocatalyst. And last to say, the conditions here, of course, are very important. We have to do this and test this in conditions which are representative of what will happen in the big tank that we saw at the beginning. Next slide, please. So just to give you an idea of the type of targets, these are some targets set here for an immobilized enzyme. They're different, of course, for a soluble enzyme or a cell, but this is for an immobilized enzyme, the gram of product per gram of immobilized enzyme plotted against the product concentration. And you can see some differences from perhaps where we begin at the very bottom left-hand corner of the slide here to where we need to get to as a target for pharmaceutical applications, fine chemicals, bulk chemicals. You can see the kind of target that we have based on the current economic situation. The price of producing protein comes down all the time. So these numbers will get more favorable, but it gives an idea about what it is we really need to achieve here. Next slide, please. To obtain some of these metrics, we can even get started in the laboratory relatively soon to make these kind of measurements. So here I show a very simple reaction you can run in the laboratory, product concentration against time. We put in 3 grams per liter of biocatalyst, we run it for 10 hours, we obtain 30 grams per liter of product, and this gives us a space-time yield. First of all, this 30 grams per liter divided by 10 hours, so 3 grams per liter per hour. It gives us a biocatalyst yield or a specific yield, as we sometimes call it. Also, this 30 grams per liter divided by the 3 grams per liter of biocatalyst, so that's 10 grams per gram. And then a product concentration we can read off directly, 30 grams per liter. Important in these kind of experiments to repeat this if there's still activity in the enzyme. So we discover everything we can get and all the activity we're able to get from the enzyme. But these kind of experiments can be done in the laboratory now. They can be done to really get us started in order to make some measurements and get an idea about how far we are from the targets which we require. Next slide, please. Beyond that, we can then think about different types of reactors. And here I show the classical 3 types of reactors we use in chemical engineering. So on the left-hand side, a packed bed reactor where we have immobilized enzyme inside. In the middle, we have here the batch reactor, which is we load up our substrate at the beginning, and over time it gets converted to product. And on the right-hand side, the continuous third tank reactor. Kinetics with enzymes don't usually favor the continuous third tank reactor. So normally we talk about either the continuous packed bed or plug flow system on the left-hand side, or the batch or fed batch system that we have in the middle. Next slide, please. So reactors like this can be modeled very easily. This was actually first done in the 1960s. And these simple mathematical descriptions here make a relationship between what kind of conversion we can expect to achieve, that's given the symbol X, for a given amount of enzyme, and that's given the symbol E. So depending on the type of conversion we want to achieve here for given substrate concentrations, depending on the properties of the enzyme, here we bring in the Km for the particular enzyme as well, the Michaelis constant, we can start to see how much enzyme we require to get to a particular level of product. So this kind of modeling is very useful to help us also to choose the correct reactor type as well. Next slide, please. Then the last part of this really concerns the process configuration. And by that, I simply mean we also need to take into account the downstream processing, how we're going to do the separation of everything, and how we're going to do the purification. The concentration that comes out of the reactor is very important for determining how easy or difficult that separation will be. So we should not forget the downstream processing part as well. Next slide, please. So just to summarize, I will say that process engineering is, of course, very important to get us out of the laboratory. But as a first step, protein engineering, enzyme engineering is really essential for us to make sure that we have an enzyme or a cell which performs at the right level we require, that gives us the metrics we require to be able to scale it up. So the procedure for moving out of the lab, as I see it, is to make economic metrics, and they're required to set some targets for the biocatalyst performance. We should try to get our biocatalyst as close to that before we start to really scale up. Otherwise, we have the challenge of trying to get a better biocatalyst at the same time as also trying to get a scaled process. The reactor type needs to be identified, and then the process configuration needs to be identified as well. And on top of that, we have the issues of scale, heterogeneities, which we will see when we start to get into a big tank, where the mixing time will become really significant relative to the time scale of the different operations we have here. I think I'll finish there, but thank you for your attention, and looking forward to the discussion later. Many thanks.
Thanks a lot, John. Thanks for joining us today and for such a very insightful presentation. I think it's very important to set also like the target of what we are trying to achieve, and taking this into consideration while you're developing a biocatalytic process. So I think it's crucial and gives a very nice overview. Thanks.
Thank you.
So let's move to our last but not least speaker, which is Albert Carceller, which is a PhD student at the Universitat Autònoma de Barcelona, which is going to give us an overview of the multi-enzymatic approach to produce lactic acid from CO2.
Okay, thank you, Annabel. So as she said, my presentation will be more focused on the lab work that we are doing for the development of one of the microbial cell factories that we are developing in the BIOCON-CO2 project, which is the multi-enzymatic synthesis of lactic acid using CO2. Next slide, please. So to present the multi-enzymatic system, as you can see in the picture, it's consisting three enzymes. The first one being alcohol dehydrogenase that converts ethanol into acetaldehyde. Then the second enzyme, which is the pyruvate decarboxylase, carboxylase this acetaldehyde into pyruvic acid. And finally, the last enzyme, the lactate dehydrogenase, produces lactic acid from this pyruvic acid. Next slide, please. We have one article published in 2011 where this system was used, and they reported to attain a lactic acid concentration of 87 micromolar. So in the BIOCON-CO2 project, in order to make it industrially feasible, we aim to reach a 22 millimolar concentration. Next slide, please. So the first thing we did is to perform the reaction as they did, but we found that the system didn't perform as expected, and we couldn't detect any lactic acid. So what we need to do is to give a more in-detail study of the system. So next slide, please. One of the first things we did was to, because we are working with CO2 in a bicarbonate buffer, was to study the influence of the pH, because the CO2 dependence of the pH of the bicarbonate buffer is pH dependent. So we've seen that changing the pH, we can modify the concentration of CO2. Next slide, please. So we also decided to test each of the reactions individually to see which one is not performing as we'd expected. In the case of alcohol dehydrogenase reaction, we seen that we could produce the acetaldehyde, but still in low amounts or low concentrations, but because the reaction is highly shifted and the equilibrium is highly shifted towards ethanol. Next slide, please. So that's what I show here. Next slide, please. Then we tested the lactate dehydrogenase reaction, which is the most favorite reaction, because it's, the equilibrium is highly shifted towards the product, which is the lactic acid. So the reaction is the one performing well. Next slide, please. Next slide, please. And finally, we got the pyruvate decarboxylase reaction, which is the one that's going to be the challenge in this project, because we first of all we have a problem with the enzyme, which is a secondary reaction that gives another product, which is called the acetoin. Next slide, please. This substrate, this side reaction was previously characterized, and it's the condensation of two acetaldehyde molecules that gives the acetoin, which is more favored than the reverse carboxylation reaction. Next slide, please. So what we need to do is to identify we identified how the system is performing. So now we need to identify which strategies can we apply to overcome these challenges. Next slide, please. So one of the first things we did is to use different variants of this pyruvate decarboxylase. We used one bacteria and then the other one from yeast. And we could observe that, for example, the yeast enzyme produces much more less acetoin, which is more promising than the bacterial enzyme. Next slide, please. And also we performed the reaction in higher amounts of CO2 by increasing the CO2 pressure. So working with reactor pressure, and also coupling the reaction to LDH in order to force the reaction to move forward and be able to detect the lactic acid. Next slide, please. So what we observed is that increasing the pressure and adding the LDH, we could shift the reaction towards the lactic acid a little bit more, and we are able to detect still in low amounts. And we could also observe that the amount of acetoin produced with the yeast enzyme makes it the candidate for the final reaction for the multi-enzymatic system. Next slide, please. We further push the reaction by adding more substrate, so we can reach up to around 74 micromolar of lactic acid. Next slide, please. And next slide, please. And we then apply these strategies to the whole multi-enzymatic system. And finally, with these conditions, we could detect the lactic acid and avoid the production of acetoin. Next slide, please. We tested different pHs, and we follow also the time course of the reaction, which, as shown here, it takes up to 96 hours, which means that the reactions are still very slow. So we need to increase the amount of enzymes. Next slide, please. And the next strategy in terms of solubilizing CO2 is the use of another enzyme, which is the carbonic anhydrase. Next slide, please. This enzyme promotes the formation of carbonic acid from CO2 and water. Next slide, please. And when we use these when we apply this enzyme with the multi-enzymatic system, because the pyruvate decarboxylase, which is the enzyme that is using CO2, prefers CO2, when we use the carbonic anhydrase, we basically see the same results without using the carbonic anhydrase. So that's not a good strategy to use in this case. Next slide, please. So what we tried, the last things we tried is to increase the amount of PDC. So in this case, we observed a threefold increase in the concentration of lactic acid, reaching up to 28 micromolar of lactic acid, which shows us that the ratio of enzyme is another big important aspect for these multi-enzymatic systems. And we will apply some designs, or we will apply some techniques like the design of experiments in order to optimize and characterize the optimal conditions for this reaction. That's all for me, and thank you for your attention. Next slide.
Thanks a lot, Albert. I think it was a nice closure. I think it's giving an idea of the challenges we are facing while trying to obtain the targeted compounds, and also it gives an overview of the combination of strategies that must be applied to actually achieve the target productivity. So thanks a lot. So I think we can just now move to the panel discussion. There have been few.
I mean, we received a few
Andreas,
questions, Annabel.
Yes.
I mean, I think two of them may be addressed to Tom. So I think one especially about industrialization. So how is the purification of phenolics done, and is it possible to scale it up to industrial scale? A yield of 95%, is it possible? Chromatographic methods often are not viable from an environmental view. So that would be one question.
Yeah, I don't know, to be honest. In the articles that this has been published in, they didn't really look in detail into the downstream processing and that kind of thing. So looking at the conversion of these molecules by the enzyme. Of course, yeah, it's a very important aspect also. I can imagine it also depends on the kind of application you're looking at. So one of the applications I mentioned for the kind of polyphenolics is as an antioxidant, for example. Then maybe it doesn't matter. Maybe you can even have a mixture. But certainly this is a next step that will have to be addressed when you have a well-working reaction system, is how do you then get the product out of it? It's certainly also a very important aspect.
And another question to John. You showed apparently one photo from a 400 cubic meter tank, which in the introduction, from which process does it originate? Maybe I missed it.
Yeah,
Okay, this is a very specific question.
That's for a process making 1,3-propanediol. So it's a microbial process, but it requires, of course, some oxygen in there. And I think that's kind of interesting, because the whole question about how to get the gas in and how to run this correctly with gases is going to be a very relevant question when we deal with CO2 as well, how to put gas into the tank. And one of the key points in a process like the one I showed was there's a very big hydrostatic head of all the liquid which is above this. So the pressure at the bottom is greater, of course, than the pressure at the top, and the residence time for the gas inside there is also important. This all goes to improving what you can get out of the gas. So I also think about how it could be in a CCU type process where we have CO2 going through the tank. And of course, we need to make sure we can get as much CO2 out of the gas which is going through this. It won't be pure CO2. So we need to really try to get the CO2 out of there and solubilize it. And I think that this having this hydrostatic head can be very helpful. That can be the type of reactor to run, like a kind of a bubble column type reactor.
Okay, thank you. I mean, Annabel?
Yeah, there is another question for Albert. It just appeared. So they ask if instead of applying pressure, would the utilization of amine for CO2 mediation would be suitable?
Sorry, could you repeat the question?
Sure. Instead of applying pressure, would the utilization of amines for CO2 mediation be suitable as an alternative strategy?
The application of?
Amines.
Amines. Sorry, what's that?
This is the technique which is used for CO2 capture today, let's say from flue gas. And I actually think it could indeed be suitable. The question a little bit is then to try to get things back out of the amines, or whether reactions can take place with other enzymes inside the amine itself. So that's a little bit the question. We change the solvent, and I think that's kind of interesting. Certainly amines are good here, but of course, if conventionally you have to get something back out of the amine, that also costs quite a bit thermodynamically and energy-wise. Yeah, so that's the challenge with using it conventionally for carbon capture. But I think it could be very interesting to explore.
Yeah. Also, maybe taking into consideration for LCA analysis to see which one will be more suitable, even in terms of feasibility.
Absolutely.
No? Okay, so well, I'll just lead the discussion then. I'll start with Marco, because I mean, I think it's one important comment you made is that, of course, like all these computational tools are helping a lot and shortening, of course, like the time, no, for obtaining the biocatalysts, which has been the main drawbacks. If we think about why biocatalysts are not really that used, because, you know, the time to develop a very efficient one, it's always much longer. Do you think that at some point, with all this machine learning and all this computational power we are achieving, will this ever replace the wet lab? My answer is no, but how long, how much do you think it could improve the, further reduce the time to develop the biocatalyst?
It's an interesting question. It's almost a philosophical question. Whether you could ever do without a lab. Well, it will take quite a while before we can close the labs, I would say. But it's certainly a very quickly moving field. The computational tools are becoming more powerful, and you really see that the time needed to improve enzymes is becoming shorter and shorter. You see these fabulous approaches now with machine learning, where you can predict structures, for example. So I do believe that there will be still great steps taken, but the labs will still be there for the next, well, let's say 20 years for sure, to validate anything you generate. I think still it's underestimated how complex even just one protein structure can be.
Yeah, that was actually something really related. I mean, we still have to rely on protein structure for having the most reliable result, which I think until we don't have a pool of structure, which is very representative, we're still far from that, no?
Yeah. Yeah, and if you just take one protein structure, it's already a challenge, but often it's not just about that structure, it also is about the interaction of that protein with other parts. If you think of biocatalysis in your medium, with solvents, for example, it's at the moment still hardly possible to include solvents in calculations. All calculations are done just based on, well, watery environment, already showing that there are certainly a lot of limitations. And I would say global characteristics can be changed now more easily, like stability, but very precise changes that are needed, for example, to change selectivity, it's still a huge challenge. Well, if you think of it, it's just changing one molecule for another for being accepted in an enzyme. So on paper, it looks simple, but no, we are still not there yet that you can engineer an enzyme from scratch. That's another example, perhaps. Engineering an enzyme from scratch, that would show that you really understand how you build an enzyme. Still not possible. It has been shown that the principle is possible, but a very effective enzyme has never been created from scratch. What has been achieved is often based on existing structures, for example, and then still you end up with an enzyme that is very lousy, very, very poor in performance. But I would say improving enzymes, there's lots of developments going on by which makes it much, much more easy than 10 years ago, or even 5 years ago. Things go fast. I would say also, except for computational developments, also the developments on technology like synthetic DNA helps a lot. The field of enzyme engineering. You can do much, much more, also experimentally, than 5 or 10 years ago. So it's an exciting field. And I think that's what you also see with companies that now are entering this field, because they also see these opportunities that you can now go for enzymes that in the past were seen as too difficult, but now perhaps are feasible.
No, absolutely. I totally agree.
Yeah.
Well, also wanted to comment from Tom. I don't know where, you've commented that biocatalytic CCU, you've explained some of the possible application. However, like the application of enzymes at higher TRL are still not really present, no? So what do you think about how do you think how far are we from applying enzymes in CCU at higher TRL? And what do you see as the main challenge?
Yes, I think there's this perspective. The selectivity and the mild reaction distance are advantages of enzymes. There are a number of challenges as well, and I think there's already been a lot discussed about enzyme engineering and how that can potentially improve the stability of enzymes. I think that another challenge that might be important when you think about really scaling up these processes is how you provide a proper thermodynamic driving force for the process in order to obtain high substrate concentrations. Because we saw in John's talk that if we want to apply an enzyme for a bulk chemical process, we have to go to substrate concentrations of hundreds of grams per liter. And the thermodynamic, let's say the intrinsic thermodynamics of many of these reactions is not very favorable. So I think that's something that needs also to have a lot of thought put into it. So how can we provide a driving force, perhaps by removing products in situ in some way? In the case of these reduction reactions, you could maybe think, how do we provide electrons in such a way that we don't have a second product form? So here maybe the electrochemistry could be interesting. But I think in addition to the development of the enzymes, this is a very large challenge that has to be addressed in the coming years if these processes are going to be industrially applied in the end.
I don't know if you, John, want to comment on that, Marco?
Yeah, I mean, I totally agree with that. Some years ago, we had looked a lot into transaminase type reactions, which we try to run backwards in order to make optically pure products. And that's exactly the type of challenge we ran into, is the thermodynamics of this. I can say there have been quite some developments in industry where people now try to remove the product, exactly as you say, in situ, but actually doing that very effectively with another enzyme. So I think that this is one of the great advantages of operating cascades, is that it can really pull the thermodynamics of this in a way better than trying to load up a lot of substrate, which, you know, Marco may tell us anyway is going to damage the enzyme. So I think it's a very good point that you make about thermodynamics. I think it's essential that we know more about it.
Yeah, I would agree on that. Marco, do you want to add something on that?
No, this is indeed also one aspect that one has to look into, and that is a very relevant one. Perhaps, there are more. That is also like costs of enzymes. So how to bring down the costs of producing enzymes. That's also often a bottleneck for a process, the costs involved. But I think also there is still room for improvement.
Yeah.
I think that the cost comes down considerably, I would say. Especially if the volume is big enough, because then we get economies of scale on making protein. So I think that's actually, there's real hope in doing that. What is much more of a challenge from my perspective is also how to measure things in the laboratory which reflect what happens in an industrial reactor. So for example, Marco may make an improvement to a protein. I may then test that under specific conditions in the laboratory, but do they reflect what really happens in an industrial process? Probably not. And then that's very hard then to predict what happens as we start to scale up. So I think that's also a kind of a challenge here. So why I do see many more processes and many more companies interested in applying enzymes, I also see some challenges there as well.
Yeah.
We have a question from the audience.
Oh, sorry.
Oh no, sorry, Annabel, I didn't want to interrupt you. Just thought, okay, anyway, we're running out of time. But if all have a few more minutes, we can just continue a bit. But I don't know. Otherwise, just let me know. I mean, we have a question. Maybe that's interesting, also from John from the audience, and also to all presenters, whoever feels concerned. When considering industrial application of biocatalysts for CCU, in your opinion, what would be more economically feasible to take advantage of the economy of scale, having big centralized biocatalytic conversion plants that receive gases from different emitting sources, or smaller conversion plants that are built directly in the different CO2 emitting industry in-house?
So that's, I think, an extremely hard question to answer, of course. In a specific case, of course, that needs to be evaluated. But what we can see is that the chemical industry is built very much on economies of scale. So operating very big scale, and of course, under those circumstances also, heavy investment is required upfront. there's been a new approach which has been suggested by a number of people, which is to come with economies of number instead, and to make things smaller scale. That takes away perhaps some risks, especially things like contamination, which can sometimes be a risk in some larger scale bioprocesses, and to try to operate then with less investment up front. And each time you make a new plant or you add a new tank, let's say, without it being so super big, you have the opportunity to make further improvements. And eventually, on average, you've made so many improvements in this that you end up with a better process than you would have had when you had to invest everything and get it right, of course, first time, when you make it very big scale. So I think it's a very interesting discussion. There's a fantastic paper in Science, I think from 2017, which discusses exactly this kind of issue. And I think it's extremely interesting debate. And I think that discussion is changing away from just huge tanks to coming into something maybe at slightly smaller scale. But it's very clear that CO2 has to match the source of the CO2 as well. So it's clear if we link this to a cement factory or to a steel works or however that is, it needs to match the scale of what we talk about there as well.
Annabel?
Yeah, there are a few other questions. Like, they appeared plenty. I don't know how much time do we want to go, like 5 minutes and then we stop? I don't know, Andreas.
I mean, I have time. We can go on for 10 more minutes. Up to you. We still have 43 attendants, so up to the panelists if they are busy with other things or have to leave.
I have another meeting I have to go to, but I can make 5 minutes. It's okay.
Okay, then let's go for 5 more minutes, maybe.
Yeah.
Sure. I think there was also a question about, can you already define a possible enzymatic reaction which could be considered to enter an industrial process? I'm not sure for whom is directed to. And I don't know if the person has like a very specific an enzymatic reaction in general. You can raise your hand or like even unmute. No?
I mean, there are, of course, several enzymatic processes operating at very big scale today, from glucose isomerase to
exactly.
different types of oxidazes. I mean, there are many.
Yeah, absolutely. Biocatalytic a lot. I think what I wanted to comment before, and then we started discussing things, was just that I think the challenge as well is that biocatalysis is clear. Like, the advantage of biocatalysis is clear when you're having a very high added value product or when you're having a very cheap enzyme. But that's why in pharma, especially when you're having more challenging product, is very well applied. I think the main issue, and I think what we should aim as well, will be like applying it for the synthesis even of like cheap compounds. That's when we know that we're reaching like a level where enzymes are as competitive as their chemical counterpart. That's my point of view.
I totally agree, Annabel. And I think one of the very interesting things when you apply an enzyme is you're not limited by the constraints of the cell. So we don't have some productivity limitations. In principle, the productivity can be very high. We have to pay a price for that, which is that we need an enzyme to be stable enough. So in a way, we are shifting from what happens with a fermentation, let's say, and we know, of course, there are quite a number of fermentations used for really big volume chemicals as well, glutamic acid, lactic acid, and so on. So I mean, just to put it a little bit in perspective, the challenge there is often about productivity. How do we really get, let's say, to 20 or even 30 gram per liter per hour? There are not very many fermentation processes that can achieve that. Enzymatic processes can certainly do that as long as they're not mass transfer limited. But now we have the issue that the enzyme needs to be cheap enough or needs to be stable enough. So I think what Marco has mentioned before is extremely important in that context.
Yeah, no, I totally agree. And just like another consideration that and then afterwards I'll leave you to the meeting and stuff. So it's just that I think it's you highlighted a very important thing, that when you scale up, you have to at some point reconsider lots of time the old process, no? Which, you know, so I think that a very important thing will be improve communication between, like, industry and who does the research to consider some stuff from the beginning, because the perspective is very different, and sometimes this communication will be the bridge to actually improve and shorten the time for developing this bioprocess, I think. So.
Yeah, absolutely, and I think sometimes it's even very challenging to be able to communicate with similar sets of units, let's say, or all sorts of things like this, which in principle should not be that hard, but somehow it turns out many times to be more complex than we might imagine, so that we're all talking in the same way and have the same end objective in mind. I think that's an important message to say, that to keep the end in mind. Ultimately, we want a big scale process, but we have to just think about that even when we're in the laboratory as well.
Yeah, I totally agree, because I think that's something that is missing sometimes, like the industry just think as the industry, the research think just as the research, and then they crash when you're trying to scale up or developing something. So I think that's just an important consideration.
Yeah, I think on top of that is that often if you talk with industry that's around already for a while, they typically think in chemical solutions and not in biochemical solutions. So, and yeah, that's often also that translates in missed opportunities. Because if you start to develop something, but you start to develop something on a chemical basis, yeah, you miss out the opportunities that you would have when you consider enzymes. And that is also still a bit of a challenge to convince companies that enzymes, or a biocatalytic process, better said, can be an alternative.
Yeah, no, totally. Sometimes, like chemists see enzymes as something that it's very scary, and they are very scared to use it. So it's biology, I don't like it, but it would be really nice at some point to just buy an enzyme as you would buy a normal catalyst and consider it in the same exact way. I think that is the challenge as well. All right, so I think we have like stolen enough time, no, to the panelists. Andreas, I don't know if you want to say something as well. I just want to thank a lot all the panelists for being here today and take time to this very nice and interesting webinar. And I want to remind that, the people assisting to today's webinar will be receiving an email for our next webinar, which will be about engineering sustainable bio-CCU. And I don't know if you want to add something, Andreas?
No, just thank you, everybody. I can see many questions not raised, not answered.
Yeah, I know. There were
more to come, I guess. It's a try, and I think also good in these COVID times that we can meet more often in webinars. Maybe too many webinars, but hopefully you consider this as a useful one. And looking forward to the next one then.
Thanks a lot.
Thanks a lot for your time.
Thank you for organizing.
All right, bye-bye.
Thank you.